My name is Vincent Tran, and I'm a postdoc in Patrick Hsu's lab at the Arc Institute. And in this video, I'll be telling you about a method we developed for rapid protein evolution. Proteins enable many functions that have been adapted for biological research and human therapies. For a given protein, we can imagine its relationship to its function as a landscape, where by changing the sequence, you can climb the landscape and enhance the function. But a protein sequence landscape is actually too large to test all possible sequences. So protein engineers have developed a wide range of techniques to efficiently determine the sequences that possess higher activity. The traditional methods for protein engineering involve making stepwise walks through the sequence space. In this framework, a set of mutations are screened on a scaffold sequence, and mutations that improve activity are kept, while mutations that reduce activity are ignored. This process can be repeated several times to improve activity. However, while this method reliably works, it fails to thoroughly explore the sequence space. So in the past decade, advances in machine learning have enabled a new set of techniques, collectively called machine learning guided direct evolution. The framework starts by experimentally testing a library of variants to train machine learning models. Then, the models are used to propose variants to experimentally test in the next round. The process is often repeated several times to robustly improve protein activity. However, from our examination of the approaches, we observed several drawbacks. This includes requiring large training datasets to train models, multiple machine learning rounds of up to 5 to 10, targeted mutagenesis of known key residues that affect function, and/or a reliance on commercial DNA synthesis to generate and test the variant genes. As a result, we were motivated to develop an approach to address all these challenges and enable rapid protein evolution. We identified 3 key components to enable rapid protein evolution. First, efficient discovery of function-enhancing mutations. Second, data-efficient machine learning to extrapolate to multi-mutants. And third, reliable lengthened variant mutagenesis to construct the multi-mutant variants to test in the lab. To enable efficient discovery of function-enhancing mutations, we drew inspiration from zero-shot methods with protein language models. We reasoned that by combining several protein language model zero-shot methods, we could increase the discovery rate of function-enhancing mutations. We developed an ensemble approach that combines both sequence-based and structure-informed protein language models and scores mutations with 2 different metrics. For each combination of model and scoring method, we then nominated a set of mutations to experimentally test. Through computational benchmarking, we showed that with the ensemble approach, we improved the discovery rate of fitness-enhancing mutations, on average, by 3-fold across different sampling sizes. So given that we can now rapidly identify enhancing mutations, we next asked how could we take these mutations and efficiently predict the optimal combination of these mutations. Previous studies have shown that neural network models enable extrapolation to multi-mutants, but relied on large training datasets. So we performed computational benchmarking to determine the minimal training dataset composition required for extrapolation. And we found the incorporation of double mutants provided a significant boost in the predictive accuracy of these neural network models through informing the models likely on the second-order epistatic interactions. And we also found that this predictive capability was maintained in lower data regimes, which confirms we can use this approach of training on just single and double mutants for data-efficient extrapolation to the multi-mutants. So now that we can predict the optimal multi-mutant combinations, we then needed a method to rapidly and reliably synthesize the proposed multi-mutants for experimental validation. We developed the method multi-assembly, which was inspired by many methods, including Darwin assembly. With multi-assembly, we made a computational design tool to design the mutagenic oligos for cloning. And in this study, we used our method to synthesize multi-mutant variants with up to 40% build efficiency and 9 mutations. Taking these 3 components, we can establish the multi-evolve framework. Our framework begins by screening single mutants, nominated from either our protein language model zero-shot ensemble approach or from existing functional data like a deep mutational scan. Then, in the second step of the framework, a set of function-enhancing mutations are chosen, and all pairwise combinations are experimentally tested. Afterwards, we train models on the compact dataset of single and double mutants, and we use the models to nominate variants. Typically, we use models to nominate the top 3 mutations at different mutational edit distances. And in this study, we test variants from 3 to 7 mutations. In the final step, all proposed variants are rapidly synthesized using multi-assembly to experimentally validate. Overall, by combining protein language models, epistasis-aware modeling, and multi-site mutagenesis, multi-evolve provides several advantages over existing methods. This includes data-efficient learning with less than 200 variants, a single machine learning round to engineer multi-mutants, and a length invariant mutagenesis method to rapidly synthesize and test the proposals. We also made a design tool to run the computational components of multi-evolve that's available at GitHub. This includes training models, designing oligos for multi-assembly, and running the protein language model zero-shot ensemble approach. More instructions can be found on the GitHub, and the software can be run using an app interface or the command line. In our study, we demonstrate the utility of multi-evolve by applying it to 3 distinct proteins. We engineer apex peroxidase and achieve greater than 100-fold improvements in activity. We applied our approach to a CRISPR-Cas protein, dCASRX, for trans-splicing, where we used data from a deep mutational scan and engineered up to 10-fold improvements in activity. And finally, we perform multi-objective optimization of an antibody to improve both binding and expression. This work was made possible through the efforts of many members in the Hsu Lab, with special thanks to Savannah Kroneman and Brian He of the ARC Institute. We're excited to see how the community uses multi-evolve. If you have any questions or want to just chat about applying this to your own proteins, please reach out. Thanks for listening.